It took me a while to figure that out. I just figured out how to do I don't like having it's really weird, but now that's like, yes. Yeah, but they're not being yeah. I saved $40. My dad wants some money.
Did you go past the instruction for like 165 on Tuesday?
No, I stayed. I saw your post on Facebook and then someone else posted and I was like, yeah, I'm staying. It's like you see
20 cars on the road, looked over, crashed into each other, and then you see all these kids standing there, they're all wearing custom sweatshirts.
No. Yeah, that's what I also was thinking. Yeah, it's gonna take so long.
And then it started really coming together, so the roads got bad. Like it was fine at first, it was 65. It's good.
All right, let's get started. So, all right, we're starting. And the first answer for the quiz is, oh, that got quiet. All right, hope you all had a good break. And so today we're going to do something a little bit different. In the past couple lectures, we've been talking strictly about catabolism or strictly about anabolism. Today we're doing both. We're going to talk about glycogen metabolism in both contexts, how we make it and how we break it and why. So we're going to first talk about glycogen breakdown, then glycogen synthesis, then regulation of glycogen degradation, and then regulation of glycogen synthesis. But the whole idea is that this glycogen is going to be helping us regulate our blood sugar. So for today, we need to review and be able to describe the structure of glycogen, and then describe the enzymatic pathways that synthesize and break down. Of course, those happy start enzymes. And then we have the regulatory mechanisms for both metabolisms, either breakdown or synthesis, and how they differ in the liver versus the muscle. Those are, again, the two main sites that we're concerned with. And then relate glycogen metabolism to maintenance of blood sugar levels with our hormones, such as glucagon, insulin, and epinephrine. So as a little review, here is our human metabolism slide. We've done everything in the blue. So we've taken sugars all the way down through the citric acid cycle, all the way through the electron transport chain, and made ATP. Now we're going to be concerned with this little tiny portion right up here and how we can take a sugar and store it for later, because sometimes in our diet we eat way too much sugar than we need on a daily basis. So there has to be a way to preserve it and keep it in our body so that in times of starvation or in times of lean eating, we have something to fall back on. So just as a review, we went over a little bit about glycogen when we talked about polysaccharides. It's a large polymer of glucose. It's about 10 to 40 nanometers in diameter. That's pretty huge considering the size of a cell. So here is a close-up of a liver cell, and all these black dots throughout that entire cell is glycogen. It's only found in the cytoplasm of certain cells, which is liver and skeletal muscle. Those are the only places that can make it. And once we break it down, the glucose can enter our bloodstream, and that glucose is free for everybody to use. But as far as making it, we can only do it in these two major sites where we also store it. So glycogen structure is pretty much just a polymer of glucose. So its glucose molecules are connected in 1,4 glycosidic bonds when it's in a straight line. However, this is a branched polymer, so there's many different branch points. These are connected by a 1,6 glycosidic bond, which happens approximately every 10 glucose molecules. So we have our 1,4s with our nice chain, and then we have our 1,6 to make our branch point. Once you have that branch point, everything past that branch is going to be the same 1,4 bonds that you would normally see in a linear polymer of that glucose. So the function of glucose differs with the different types of tissues that it's used in. So in the liver, you're going to both synthesize and break down glycogen, and that's really related to your maintenance of blood sugar levels, your blood glucose levels. The muscle, however, synthesizes and breaks down glycogen to regulate energy requirements of the muscle cell. Again, if you're doing a lot of strenuous exercise for a prolonged period of time where you're not really eating in between, this is when you're really going to be thankful that you have that glycogen stored up in your muscle because it allows you to keep going. So first we'll talk about glycogen breakdown. So this is when you want to utilize that glycogen, break off bit by bit pieces off of that glycogen to make glucose and either use it in your skeletal muscle immediately or send it into the bloodstream to regulate your blood glucose level. So with glycogen, you start off with this long polymer and you're eventually going to just chip at it one piece after one piece after one piece, just removing one piece of glucose at a time. You do this in a multiple step process where first you remove one glucose 1-phosphate using an enzyme called glycogen phosphorylase. You then will turn that glucose 1-phosphate into glucose 6-phosphate. And if you remember from our glycolysis lectures, glucose 1-phosphate is stuck inside of cells. You can't do anything with it unless you're interested in just its use in liver or skeletal muscle. Glucose 6-phosphate, however, can be used elsewhere. It can go through glycolysis. It can be turned back into glucose in the liver by glucose 6-phosphatase, and then glucose is transported out into the blood for use by multiple tissues. Or it can go through a pathway we're going to talk about on Wednesday called the pentose phosphate pathway, where we can actually make ribose and NADPH. This is a pathway we'll just talk about later, so we'll ignore that for today. But this glucose 6-phosphate, the reason why it can enter directly into glycolysis is because glucose 6-phosphate is one of the intermediates of glycolysis. So you already primed it. You can skip a step and just put it right in and get your pyruvate, your lactate, or from pyruvate down to CO2 in water, and of course ATP. So important, the first step is glycogen phosphorylase will catalyze this breakdown. This is, again, not one of those start enzymes, just this is how we start, is that we have glycogen. We're going to add a phosphate group and attach it to one of those glucose molecules on the very end of glycogen. So glycogen is left whole at just one sugar less. That's why it's N-1 right here as a subscript. You end up with this glucose 1-phosphate. And so this is a phosphorylysis reaction, which is kind of unique because usually phosphorylation, you just add a phosphate group. This is you're adding a phosphate group and you're breaking something. So it's kind of unique. And it actually does not require ATP. This is inorganic phosphate that's just floating around. No catalysis of ATP is needed. And the glucose 1-phosphate is released, but no transporter in cells, so it can't leave the cell. So if it's the liver, it's staying in the liver. It's the muscle, it's staying in the muscle. So now we have this glucose 1-phosphate, and then we're going to come to the starred enzyme, glycogen phosphorylase. This is a dimer of 2 identical subunits, so there's actually 2 of these enzymes connected together. So it's very efficient in that it has 2 catalytic sites, so it can do 2 times the reaction at the same speed. It also has some important allosteric sites that are important, which we'll talk about soon, later in this talk. It can bind also to glycogen, and it will use the site to perform a cleavage reaction. So we're going to be breaking things. It uses an interesting type of cofactor called pyridoxal phosphate. It's abbreviated PLP. It's a derivative of vitamin B6. And we'll see how this PLP works with the enzyme. So this glycogen phosphorylase, the PLP is just going to be represented here with this blue and just the R. The important part is it has this phosphate group attached to it. It's a rather large molecule, so we're just going to abbreviate it with that R. these two phosphates are going to interact with that glucose 1-phosphate. It forms a carbocation intermediate, which is also pretty unique because typically carbons don't form cations or anions. They're usually charged neutral. But here we have a plus charge on this carbon, and it will accept the phosphate group, and it will essentially move a phosphate. So the energy is breaking the alpha 1,4 linkages that are stored in the carbocation. And so once we have all of these little pieces chipped off, we're going to basically chip off one glucose after one glucose after one glucose, and you move along this linear line just in progression. Just one glucose removed, one, just take them off. But eventually, you're going to be coming to a branching site. Now, all of these enzymes have been working at an alpha 1,4 linkage. However, those branching sites are alpha 1,6 linkages. So naturally, those enzymes aren't going to work. So we have to do something else so that we can actually utilize the glucose that are held together by this branch point. So what we'll do is we remove those glucoses up into a point where we're about 4 glucose molecules before the branch. So just these molecules here. We then use an enzyme called a transferase, and we essentially will remove 3 of those glucoses to the next linear chain. So we're going to just shift them. And that leaves us with only one glucose that is connected to that branch point. And then there's an enzyme that will hydrolyze that one glucose off, and only that one glucose. So in that way, it's a little bit more restricted in its substrate. It will only cleave that alpha 1,6 linkage. So you just have to clear the way so that it actually can come in. So this glucosidase will then cleave off that branched point, that 1,6 linkage, and then that will be free. And now you're left with all that rest of that nice linear piece of polymer. Now remember, these glycogen molecules are huge. There's many and multiple of these branch points. So it's very important that we do this, and it happens regularly. It's not something infrequent, because almost every 10 glucose molecules, there's going to be another branch point. So all of these enzymatic activities that we talked about in transferring those glucoses and then breaking that 1,6 glucosidase bond, that's all done by one enzyme that has 2 activities. And it has this nice name that we can really easily remember, the debranching enzyme. That's exactly what it does. It just takes the branch right off, where it does the transferase activity, so it removes those 3 glucose molecules. It puts it right onto that core linear line of a glucose polymer in the glycogen, and then it's going to cleave off that alpha 1,6 linked glucose. It does both these functions with one enzyme. It's very efficient. And it's also very easy to remember that name. So once we have these glycogen, essentially this glucosidase, this debranching enzyme, is going to use water to cleave it in 2, so that you will break that 1,6 linkage, and you're left with a glucose, and you're left with glycogen, just with one less residue on top of it. Same thing. The end product is the same as if it was just the normal way of removing one glucose. So the main thing to remember is glucose 1-phosphate is a major product of glycogenolysis, so breaking up of glycogen. So we have glucose in this polymer of glycogen. You add that phosphate group, you have glycogen, and you have this glucose 1-phosphate. However, glucose 1-phosphate cannot directly enter into glycolysis. There's no intermediate of glycolysis that's glucose 1-phosphate. So if you want to use it for glycolysis, you're going to have to convert it. So to convert it, we just convert it to something that's actually in glycolysis, and that's glucose 6-phosphate. So you go from glucose 1-phosphate to glucose 6-phosphate. We're essentially just moving the phosphate around. This is done by an enzyme called phosphoglucomutase, where we have a phosphate on a serine inside of the enzyme, and then we attach it onto our glucose 1-phosphate at the 6-carbon position, and then we cleave off that phosphate on the first carbon position and put it right back onto that enzyme, so we regenerate our catalyst, and we have our glucose 6-phosphate. This is an intermediate of glycolysis. It can enter right in and go through glycolysis. So that would be, again, glycogen, glucose 1-phosphate, glucose 6-phosphate, enter through glycolysis. Of course, that can happen. The glucose 6-phosphate can go through glycolysis. We use this in a lot of tissues like muscle and brain that needs energy fast. These are highly energy dependent muscles and organs that rely on a lot of glycolysis because of their high energy demands of neurons or of muscle cells. In the liver, oftentimes we don't go through the glycolysis pathway, but we're going to be going through and turning it back into glucose to release into our tissues to regulate blood glucose levels. And that's one of the main functions, of course, of the liver, is to respond to our blood sugar levels and then adjust accordingly. Because remember, the liver not only is breaking and making glycogen, but it's also doing gluconeogenesis as well. So it has a lot of regulation of glucose and sugar roles for the liver. So glucose 6-phosphate is turned into glucose by a specific enzyme called glucose 6-phosphatase. So basically, you're just cleaving off that phosphate on the glucose molecule, you produce glucose, and this is only present in the liver, but not in the muscle. This glucose is released into the blood, and it can be taken up by the brain or active muscles or wherever it's needed in response to that low blood sugar level. So we'll have a quick review question. So why is glycogen phosphorylase insufficient for mobilizing all glucose residues from glycogen? So we have majority of the class choosing choice C. Has everybody had a chance? Everybody done? We have a little bit, at one point it's higher, of the choice B, glycogen is helical and requires unwinding. So the correct answer is, of course, C, which the class chose as the majority. Glycogen is branched, and we need specific enzymes to actually take care of that branching. That debranching enzyme is going to take care of that branch point because the glycogen phosphorylase cannot do that. It can only break the 1,4 links. What was interesting is that the other choice at one point did have a lot of, had some students suggest selecting that one, is that it's a helical molecule and requires unwinding. Now, it is a helical molecule. A lot of these branches are intertwined within each other, and that's something we mentioned when we talked about the structure of glycogen a couple lectures ago when we talked about sugars. However, unlike DNA, you don't have to unwind glycogen to break it apart. You just have to use these enzymes to chip off that one glucose at a time. Because you're doing it as a sequential, 1 unit at a time type of measure, it actually doesn't require that unwinding. The enzymes really don't seem to care about that kind of thing. Whereas DNA, you have to unwind it to replicate and things like that. So it was an interesting choice. So glycogen synthesis, we've now broken it. Now let's try and make it. So glycogen synthesis starts off with basically going a little bit in reverse. So we have glucose, we're going to use some ATP, and then we're going to produce glucose 6-phosphate. And this is done by different enzymes depending on which tissue you're in. If it's in the muscle, it's done by hexokinase, and if it's in the liver, it's done by glucokinase. Of course, hexokinase is an enzyme we should be familiar with, being involved with glycolysis. Now, the glucose 6-phosphate is then turned into glucose 1-phosphate, which is done by an enzyme called phosphoglucomutase. If we just look at the next step, keep going forward, we have glucose 1-phosphate interacting with a activated molecule UTP, which is like ATP, but it's uridine and not adenine, so it's UTP. And this is done by an enzyme called UDP glucose pyrophosphorylase. Again, this isn't starred, so it's not important to remember that one's name. But we're essentially going to be adding a UDP onto the glucose 1-phosphate to produce UDP glucose. Now, the UDP glucose then is used to add one glucose molecule onto glycogen by an enzyme, which is starred, glycogen synthase. Pretty easy to know the name of, yes.
So, quick question. The UDP glucose, or in general, if it's not starred, what should we know about it?
So,
everything except the name? You
don't need to know the name, no. What you need to know is something like this, glucose 6-phosphate. You don't need to know what did it. You need to know how are we starting in the general flow. So we're just going to glucose 6-phosphate, glucose 1-phosphate. Now we're going to glucose 1-phosphate. The UDP glucose comes next. And now we're just going to add that onto glycogen to make the glycogen just one glucose longer. So this all takes in consideration that you have this glucose polymer already started. So here is just how we add one glucose on at a time. So we're left at the end of this with UDP, which can cycle back and form UTP, and then the whole process can repeat. And then we have glycogen with one residue longer, so N+1 residues. So just like when we broke down glycogen, step by step, one glucose at a time, that's how we build glycogen, one glucose at a time, one at a time in this polymer. However, as I said, this all relies on you already having glycogen there. So what happens when your cells divide? You have one liver cell to two liver cells. Do those liver cells have glycogen to begin with? This has to happen at some point. You're not just born with glycogen. Glycogen has to be produced at some point to add more glucose onto. So there has to be some way to start the whole process. So at the very beginning, we have a protein, which is different, because now we've been all talking about sugars. But now at the very beginning of glycogen synthesis, we have a protein called glycogenin. And this is a rather large protein, 37 kilodalton protein, that is glycosylated on a specific tyrosine residue. And from this one tyrosine residue glycosylated, meaning just put this glucose on that tyrosine, we're going to build off of that to produce our glycogen. So here in the picture, the glycogenin is just represented by that G in the circle. And then you have glucose built off of it. And then, of course, you have these branch points. And then you have more polymers with more branch points. It just gets bigger and bigger and spider webs out of there. So it becomes this large molecule, all from this one protein, which has that glycosyl group on the tyrosine. Now, the glycogenin itself is an enzyme, and it will catalyze the formation of 7 glucose long chain onto itself. However, once it's finished with that 7 glucose chain, it won't do anything else. It's just kind of just like the seed of which you start this large molecule to grow off of. Once that 7-member chain is done, it's no longer going to be in use. The glycogen synthase and all the other enzymes are going to just take over to make that chain even longer. So this is how, using that glycogen synthase off of that glycogenin, we're going to produce that first initial chain. We're going to produce 7 glucoses with the glycogenin, and then off of that we're going to add additional ones with the glycogen synthase. Now what happens when we need a branch point? Again, these are a branched molecule. Glycogen synthase and glycogenin are only producing a linear polymer. So we have to produce those branch points to make that nice structure of glycogen we've been talking about. So now we're going to use an enzyme aptly named the branching enzyme. So whoever discovered a lot of these enzymes like to keep it simple. The branching enzyme, synthetase, just they kept it nice and easy, which is something you appreciate. So the branching enzyme will basically, you have this long chain of, linear chain of glucose. We're going to take off 1, 2, 3, 4, 7 of these glucose molecules, and then we're going to move it onto an earlier glucose and form that 1,6 linkage. So we're just going to make one long chain into 2 chains that are attached by a branch. And so now we have these two ends here, which glycogen synthase can come in and add additional glucose down and make those two chains longer and longer until you need another branch point. And then the branching enzyme will do its work all over again. So just by forming these branches, we can then make that very large molecule out of that one very small, that one protein that we began with. So as a review question, glycogen synthase attaches glucose molecules through a formation of 1,6 glycosidic bonds. True or false? Ooh, almost 50-50. Okay. Still moving, okay. Everyone have a chance to answer? All right, so we have majority of the class saying false, and then about a third of the class saying true. So it is false. So the glycogen synthase attaches molecules in a 1,4. So this is the enzyme, the synthase, that's going to make the long chain polymer. The branching enzyme is what's going to do those 1,6. So the 1,6 glycosidic bonds are only present at that, between the two glucose molecules that forms the branch. All those other molecules of glucose are connected through those 1,4. So glycogen branching enzyme is going to do the 1,6. The glycogen synthase is going to do the 1,4. So we've spent most of the class talking about how we break it. Now we've talked about how we've made the glycogen. Now let's actually talk about how we utilize it. And so talking about how we use the glycogen is really talking about how we regulate it, because we don't want to form glycogen all the time. We'd use up all of our sugar stores. We'd use up all the sugar in our blood, basically, making this glycogen. We don't want to break it down all the time, because then our blood glucose levels would go way through the roof, and everyone would end up with like diabetes or something. And so we need to be able to properly regulate the equilibrium of making glycogen and breaking glycogen. So we'll first talk about how we regulate the degradation of glycogen. So when we're breaking down glycogen, how do we regulate it? And it's mainly at that phosphorylase level. So that's one of the reasons why that enzyme is starred, is because this is the enzyme at which we do all the regulation when we break down glycogen. And we do it either through phosphorylation or allosteric modulation. So we'll compare and contrast what regulates glycogen metabolism in our two tissues of interest, our muscle and liver. So in the muscle, we're often using glycogen to respond to times of strenuous exercise or strenuous running or things like that. Think of like the fight or flight response. So it's the use of glycogen, the breaking down of it, is triggered by the release of epinephrine, one of our hormones. Where in the liver, the rapid mobilization of glycogen is triggered by glucagon, which is a peptide hormone secreted from the pancreas, and it responds to low blood glucose levels. So when blood glucose levels are low, glucagon comes out, goes into the bloodstream, taken up by the liver. The liver says, hey, let's put out some more glucose, and then we start to break down our glycogen. So both epinephrine and glucagon are secreted into the blood and will interact with specific receptors on the surface of the liver or muscle cell. This is kind of generalized to talk about both. So the exact names of the receptors, of course, are not that important right now. But what happens is that it's a G protein coupled receptor, which will cause adenylate cyclase to create cyclic AMP from ATP, which will activate protein kinase A, which will then activate phosphorylase kinase, and then will then phosphorylate your phosphorylate the phosphorylase. It's a little bit of a mouthful. So when the glycogen phosphorylase is phosphorylated, it is now active. So that is how phosphorylation will regulate it. It's all due to the sensing of those secondary messengers coming down through those epinephrine or glucagon levels. You have these hormones sensed by these cells. We have the secondary messengers coming down, we get phosphorylation, and then we get an active glycogen phosphorylase. Now, I also said that this enzyme is modulated by allosteric regulation. So if we remember, allostery is basically we have some type of effector molecule which binds in a site that is not the active site, and then we'll shift whether the enzyme is active or inactive. The T state is the less active, which is can be stabilized by inhibitors, and the R state is more active, which is stabilized by substrates or by activators. And so you shift between the two. Now, when we talk about the glycogen and how it gets metabolized, it's going to be different in the muscle and the liver. So we'll first start with the muscle. Now, going back a little bit, we said how this glycogen phosphorylase is a dimer of itself. So there's two of these units. So that's why it's represented here, where it's two exactly the same molecules just stacked one on top of each other. Each one has nucleotide binding sites, so it can bind to AMP. So when you have AMP binding here, it will glucose 6-phos will stimulate that R state, so it makes it more active. And so you can see that there's this branch inside of the enzyme that when the modulator binds, it will actually cause a conformational change and take that part of the polymer out of the way, basically, because these are the active site here for the for its substrate. And by having these allosteric modulators come in, it removes this blockade so that the substrate can come in and bind. Not only does AMP stimulate the R state, but glucose 6-phosphate and ATP stimulates the T state. So when you're under low energy conditions, you want to release glucose. So the AMP activates this enzyme. However, if you have high energy demand, you have high glucose levels, you have this glucose 6-phosphate or ATP coming in, it'll shunt that R state back over to the T state. Now in the muscle, we're usually in default mode in this T state. It's really under those energy poor conditions. So when you're having the fight or flight response, when you're doing this long exercise, if you're running a marathon, that kind of thing that you need to chew up all that glycogen to give your cells the energy that it needs, that's when you're going to shift it over to being the more active R state. So muscle, you tend to be in that T state. So this gets a little bit more tricky because we also talk about how it's phosphorylated. So there are phosphorylated states and there are allosteric activated states. And again, default here is the inactive protein inside of the muscle where there's no phosphorylation and there's no allosteric modulators. Epinephrine, remember fight or flight, it will signal a cascade that leads to phosphorylation, and this phosphorylase kinase will add that phosphate group. And when phosphorylated, it becomes active, so it has its little a by it, whereas inactive, it's often called little b. So we have all these different modulators coming in. We have the most active state is here at the upper right, where you're both phosphorylated and you have your allosteric modulators at AMP coming in, and it's the most active, and under the right conditions, such as strenuous exercise, fight or flight, you're going to have that. When you're in the lower energy state, you're going to be here in default, just at your everyday walking, just sitting down, you're not going to need that glycogen being broken down. You're going to be stuck here in that T state for the enzyme in the muscle. Now, there are the combinations of the two where you have either allosteric modulator or the alone but not the phosphorylation, or you have the phosphorylation but not the allosteric modulator, which are essentially these two here at the, this diagonal. They're somewhat active but not as active as this most actively labeled one here. So your ultimate goal to have the proper regulation of glycogen, to have the most activity of this enzyme, is to have both the allosteric modulator present and the phosphorylation present. Of course, once you have this phosphoryl group on there, once you stop all that strenuous exercise, you don't need it to be active anymore, so you need to remove that phosphate group. So that's done through a protein phosphatase called protein phosphatase 1. Phosphatases are just enzymes that remove a phosphate group. So you remove the phosphate group and you can put it back into a lower activity. So to summarize, we start in the T state, unphosphorylated in our muscle. We have all of our signals, all of the right conditions with our allosteric modulator, our phosphorylation. We make it most activated, so it's phosphorylated. We have our allosteric modulator in there, and then we will get our glycogen broken down. We then have to remove that phosphate group to turn it back off, to bring it back to the T state. So this is, again, only in the muscle, these conditions. In the liver, it is different. In the liver, glucose levels also act as an allosteric modulator and will stimulate the T state. So if you have high levels of blood glucose, you don't need to break down your glycogen anymore to make more glucose because you're fine. So glucose will come in and will then allow for that T state to form. And usually with the liver, you're in default under the phosphorylase A, so the activated condition. So you really need that blood glucose level to turn it off. Remember, in the muscle, the default is the T state. In the liver, the default is the R state. So again, when you have these, when you have the activation of the phosphorylase, you also want to turn off it with this glucose, using the protein phosphatase. You remove those phosphate groups and you send it back into that T state. So you remove it off, which is kind of what these purple Pac-Mans are coming in here to chew up that, that phosphate group. So again, glucose, high levels in the blood, you want to turn off your glycogen metabolism, turn off that phosphorylase A by coming in, binding into the allosteric site. It shifts from the R to the T state, and the phosphatases come in, remove those phosphate groups, and then take off the, keep that enzyme off, essentially. So importantly, we know that we have reciprocal regulation of glycogen metabolism. So if you have your signal of either glucose, glucagon, or epinephrine coming in, transmitting through, we talked about how these secondary messengers can turn on the phosphorylase by going from the inactive to the phosphorylated active form. At the same time, when you have this signal to break down your glycogen, you don't want to also be creating glycogen. It would just be sending mixed messages if we need to break it, we need to make it all at the same time. So while at the same time this protein kinase A will activate your glycogen phosphorylate, it also inhibits your glycogen synthase. So by inhibiting that glycogen synthase, you're going to be turning off that synthesis. So you're not doing two things at once. You're only going down one route. You're going to be making your you're going to be breaking your glycogen. You're not going to be making your glycogen. Is that clear? All right, so now we'll shift over to how do we regulate the synthesis of glycogen. So, excuse me. With glycogen synthesis, it's usually a coordinated regulation, so that if you were to look at enzymatic activity over time, this is all in a very quick scale, you'll notice. It's a matter of minutes. If you add glucose, your synthase activity will rise very shortly. Within 4 minutes, you start to have a lot of activity of that synthase. So you're going to be making your glycogen. At the same time, that phosphorylase, which inhibits, which breaks down that glycogen, that enzymatic activity decreases very quickly as soon as you add that glucose. So this coordinated regulation is very important. And so we've talked about this phosphorylase, how we break down that glycogen, how that's regulated in response to glucose, for example, in the liver. Now let's talk about how we will synthesize it and make our glycogen. So both the liver and the muscle both respond to insulin as the signal for making glycogen. Again, for breaking down glycogen, they had two different signals. It's the glucagon or epinephrine. But to make glycogen, we're going to use the same hormone, which is insulin, which is, of course, secreted by the pancreas under conditions of high levels of glucose. It'll bind its receptor. This is a different kind of receptor. It's a kinase type receptor, which will then cause a phosphorylation of many different proteins, one of which is it will phosphorylate, one of these kinases will phosphorylate this glycogen synthase kinase, which takes it from an active state into an inactive state. And this glycogen synthase kinase is what will phosphorylate glycogen synthase. And then it will cause this different type of regulation that is seen here. So the phosphorylation events inhibits the glycogen breakdown and inhibits glycogen synthase. And inhibits glycogen breakdown and inactivation of glycogen synthase. However, the protein phosphatase 1, our same character from before, will remove the phosphate from glycogen synthase and will activate it. So there are these two different signals going on, where we have the phosphatates activating the synthase, and we have the kinase inactivating it. And it's all under which condition that you're under. So let's explore that a little bit more. So the protein phosphatase 1 removes phosphate from glycogen synthase, which will activate it. So you start with a meal, and or at rest. Glucagon or epinephrine comes in, will activate protein kinase A, and inhibits our glycogen synthase over here at the same time. However, at the same time, glycogen synthesis is needed at some point. So we have this protein phosphatase A, which will then convert and take off that phosphoryl group, and then we will then lead it over to an activated form for the glycogen synthase. At the same time, the protein phosphatase 1 will remove the phosphate from the phosphorylase kinase and inactivate it so that you're not having two signals going on at the same time. So we can look at it reduced down to this little scheme, which is, for me, is much easier to visualize and much easier to follow. So under fasting glucose levels, your pancreas will release glucagon, and the liver glucagon receptors will take those up, and the liver will then go through all the breakdown of glycogen to release glucose into the blood. That's on one hand. However, when you're under the you're in the fed state, you have high blood glucose levels, your pancreas will release insulin. The body cells will then have these insulin receptors, and then the body cells will take up glucose from the blood. And then we have the balance of both sides so that you try and maintain that blood sugar level. Of course, when those levels get changed with something like diabetes, these all will change because it tips the balance, and you end up with a disease condition rather than the healthy everyday state. So to review, Glycogen is the major source of storage polysaccharides in mammals. Both synthesis and degradation are coordinated processes that occur only in the liver and the muscle. Both synthesis and degradation of glycogen are regulated by allosteric binding and by phosphorylation and different enzymes. And blood glucose levels, of course, are modulated by hormones. So they help us to know whether we should be making glycogen or breaking it down. And again, those hormones are all related to conditions. Are we in a fed state? Are we in a fasting state? Are we going under fight or flight response? Are we in a marathon? Are we using so much energy that we can't keep up just with glycolysis? We need an extra boost, that kind of thing. So those hormones will tell the muscle or the liver what to do and how to do it, basically. Questions about glycogen? I know it's a lot of information in this one lecture, it is, but that's what we have for today, yeah. Okay, that is all for today. Again, we have the quiz on Wednesday, and we'll look at the final fate of glucose, which is at where we turn glucose into ribose and NADPH. And then we'll be done with glucose for a while.